Unit 3. Molecular Biology. Sits at the heart of every CSIR NET Life Sciences examination. It covers the central dogma: DNA replication, transcription, translation, gene regulation, RNA interference, and recombinant DNA technology. Questions from this unit appear in every section. And Section C questions almost always require you to reason from a molecular mechanism to an experimental outcome, never just to recall a definition. This video selects 5 questions from the CSIR NET 2016 examination. Each one covers a different domain of molecular biology. And each one teaches a reasoning framework that transfers directly to every other question in that domain. The 5 topics are: the lacZ operator mutation, DNA polymerase III proofreading, lagging strand synthesis, siRNA-mediated gene silencing, and gene cloning with insertional inactivation. Master these 5 mechanisms, and you will be equipped to answer any variant the exam produces. Let us begin. Question 1. In the Jacob monod model of the lacZ, a mutation in the operator that prevents repressor binding would result in: Option A, increased sensitivity to repressor. Option B, catabolite repression only. Option C, no lac gene expression. Option D, constitutive expression of lac genes regardless of lactose. Before reading on, think about what the operator actually does in the lacZ. The repressor binds the operator to physically block RNA polymerase from transcribing the structural genes. If the operator is mutated and the repressor can no longer bind, what happens to the lac genes? Are they now always on, always off, or controlled by something else entirely? The correct answer is option D. Constitutive expression of lac genes regardless of lactose. The lacZ has 2 regulatory mechanisms working in parallel: repressor operator control and catabolite repression through CAP and cyclic AMP. The repressor protein, when there is no lactose present, binds the operator and prevents RNA polymerase from transcribing lacZ, lacE, and lacA. When lactose enters the cell, allolactose, its isomer, binds the repressor, causing a conformational change that releases it from the operator. Transcription can then begin. Now consider the mutant operator. The mutation prevents the repressor from binding under any condition, whether lactose is present or absent. The physical block is permanently removed. RNA polymerase has unobstructed access to the promoter at all times, and the lac genes are transcribed constitutively, that is, continuously, regardless of whether lactose is available. Option A is wrong. A mutant operator that cannot bind repressor cannot show increased sensitivity. There is nothing to be sensitive to. Option C is the exact opposite of what happens. Constitutive mutation leads to maximum expression, not zero. Option B is a partial truth that misses the point. Catabolite repression still functions because it acts at the promoter through cap binding, not through the operator. But catabolite repression is a separate regulatory layer. The question asks specifically about the consequence of preventing repressor binding at the operator. Framework. Whenever you see an operator mutation in a CSIR NET question, ask immediately, does this prevent repressor binding? If yes, the result is constitutive expression. Question 2. The mechanism of proofreading by DNA polymerase III in E. coli involves: Option A, recognition of mismatches by a separate repair enzyme. Option B, 3' to 5' exonuclease activity of the epsilon subunit that excises misincorporated. Nucleotides. Option C. RNA primer replacement. Option D. UV light detection. Pause here. Proofreading is a co-replicative process, not a post-replicative one. Think about what structural component of the polymerase itself would allow it to detect and remove a wrong nucleotide the instant it is incorporated. The correct answer is option B. 3' to 5' exonuclease activity of the epsilon subunit. DNA polymerase III is a multi-subunit holoenzyme. The alpha subunit carries the polymerase activity and synthesizes DNA in the 5' to 3' direction. The epsilon subunit, encoded by the DNA Q gene, carries the 3' to 5' exonuclease activity that makes proofreading possible. Here is the mechanism. When a wrong nucleotide is incorporated during synthesis, the mismatch destabilizes the nascent 3' end. This prevents the alpha subunit from adding the next correct nucleotide. The 3' end shifts into the active site of the epsilon subunit. The epsilon subunit then cleaves the phosphodiester bond and excises the mis-incorporated nucleotide. The correct nucleotide is then incorporated, and synthesis continues. This proofreading mechanism increases replication fidelity by approximately 100 to 1,000-fold beyond what base pairing selectivity alone achieves. Option A describes a different process entirely. Mismatch repair, carried out post-replication by the MUTS, MUT-L, and MUT system. Mismatch repair corrects errors that escaped proofreading. They are not the same mechanism. Option C describes RNA primer replacement. The 5' to 3' exonuclease activity of DNA polymerase I, which removes RNA primers and fills the gaps. Option D has no basis in E. coli replication biology. Framework. Proofreading equals 3' to 5' exonuclease equals epsilon subunit. Mismatch repair is post-replicative and uses MUTS MUT. Never confuse the two. Question 3. In eukaryotic DNA replication, lagging strand synthesis requires Option A. Only DNA polymerase epsilon. Option B. Topoisomerase II as primase. Option C. A single continuous RNA primer. Option D. Multiple Okazaki fragments, each initiated by a new RNA primer. This question tests your understanding of the structural asymmetry of the replication fork. Both strands of the parental DNA are replicated simultaneously as the fork advances. One strand, the leading strand, runs in a direction that allows continuous synthesis toward the fork. The other strand, the lagging strand, runs in the opposite direction. Think about what this means for how the lagging strand must be synthesized. The correct answer is option D. Multiple Okazaki fragments, each initiated by a new RNA primer. The anti-parallel nature of DNA creates the fundamental problem of lagging strand synthesis. DNA polymerases can only synthesize in the 5' to 3' direction. The leading strand template runs 3' to 5' toward the fork. So DNA polymerase can follow the fork and synthesize continuously. This produces the leading strand in one long uninterrupted chain from a single primer at the origin. The lagging strand template runs 5' to 3' toward the fork, meaning DNA polymerase must move away from the fork to synthesize in the correct direction. Synthesis must therefore proceed in short discontinuous segments. Okazaki fragments, each approximately 100 to 200 nucleotides long in eukaryotes, and 1000 to 2000 nucleotides long in prokaryotes. Critically, Each Okazaki fragment requires its own RNA primer. The primase enzyme synthesizes a short RNA oligonucleotide, approximately 10 nucleotides, to provide the 3' hydroxyl that DNA polymerase delta needs to begin extending the fragment. After synthesis, the RNA primers are removed by the flap endonuclease 1. The gaps are filled by DNA polymerase delta, and the fragments are joined by DNA ligase. Option A is wrong. DNA polymerase epsilon synthesizes the leading strand in eukaryotes, not the lagging strand exclusively. Option B is nonsense. Topoisomerase resolves supercoils ahead of the fork. It has no primase activity. Option C is the leading strand, not the lagging. Framework. Lagging strand equals Okazaki fragments. Okazaki fragments equal multiple RNA primers. One fragment equals one primer from primase. Question 4. RNA-induced silencing complex loaded with siRNA cleaves target mRNA at. Option A. The 5' cap site. Option B. The position base paired with nucleotides 10 and 11 of the guide strand, catalyzed by Argonaute. Option C. The polyadenylation signal. Option D. The AUG start codon. Pause here. The siRNA is a 21 nucleotide double-stranded RNA with 2 nucleotide 3' overhangs. One strand, the guide strand, is loaded into RISC and base pairs with the target mRNA. Think about where, within that 21 nucleotide duplex, the catalytic subunit of RISC would make its cut. The correct answer is option B. Cleavage occurs at the position base paired with nucleotides 10 and 11 of the guide strand, catalyzed by Argonaute 2. RISC. The RNA-induced silencing complex is built around the Argonaute protein family. In the siRNA pathway, Argonaute 2 is loaded with the guide strand of the siRNA duplex. The guide strand has perfect complementarity to the target mRNA. When Argonaute 2 binds the target through this complementarity, it positions its catalytic PYW domain, which has RNase-H-like endonuclease activity, over the target mRNA backbone. The cleavage site is precisely defined. It falls between the nucleotide complementary to position 10 and the nucleotide complementary to position 11 of the guide strand, counting from the 5' end of the guide. This places the cut at the center of the 21 nucleotide guide target duplex. The cleavage produces a 5' phosphate and a 3' hydroxyl on the target mRNA fragments, which are then rapidly degraded by cellular exonucleases. The precision of this cleavage is the molecular basis of RNA specificity. Option A is wrong. The 5' cap is at the very terminus of the mRNA and has no relationship to the siRNA guide sequence. Option C is wrong. The polyadenylation signal is at the 3' end and is a sequence feature unrelated to RISC targeting. Option D is wrong. The AUG codon is a sequence in the coding region. Argonaute does not target it unless the guide strand happens to pair there. Framework. RISC cuts between positions 10 and 11 of the guide strand. This is the universal rule for siRNA-mediated cleavage. Question 5. In a typical gene cloning experiment, by mistake, a researcher introduced the DNA of interest within the ampicillin-resistant gene instead of the lacZ gene. Which of the following would be the result? Option A. The bacteria which took up the plasmids would grow and give blue colonies. Option B. The bacteria which took up the plasmids would not grow. Option C. The bacteria which took up the plasmids would form white colonies. Option D. all of the bacteria would grow and give white colonies. Pause here. Standard blue-white screening works because the insert disrupts lacZ, preventing beta-galactosidase production. So colonies are white instead of blue on X-gal plates. But this researcher has put the insert in the wrong gene. Not lacZ, but the ampicillin resistance gene. Think through what happens now when the bacteria are plated on ampicillin-containing medium. The correct answer is option B. The bacteria which took up the plasmids would not grow. To understand this, you need to trace the logic of antibiotic selection in cloning experiments. The plasmid carries two key elements: an antibiotic resistance gene, here, the ampicillin resistance gene encoding beta-lactamase, and the lacZ gene for blue-white color screening. Selection on ampicillin plates ensures that only bacteria that have taken up the plasmid survive. Bacteria without a plasmid die on ampicillin. Now, the researcher has introduced the insert into the ampicillin resistance gene, not into lacZ. Insertional inactivation of the AmpR gene means the bacteria cannot produce beta-lactamase. Beta-lactamase is the enzyme that degrades ampicillin. Without it, the cells cannot survive in the presence of the antibiotic. When plated on ampicillin medium, these bacteria, even though they carry the plasmid, are killed. They do not grow, and therefore no colonies of any color appear. Option A, blue colonies, would require functional AmpR and functional lacZ. Here, AmpR is disrupted. Option C, white colonies, is the standard result of a successful cloning experiment where the insert is in lacZ with functional AmpR. Here, the selection fails at the first step. Option D, all bacteria grow, is impossible without an intact antibiotic resistance gene. Framework. Always trace the selection logic first before considering the reporter. If the selectable marker is disrupted, the bacteria die. Color is irrelevant. Let us consolidate the 5 reasoning frameworks from this video. Framework 1. A lac Operon operator mutation that prevents repressor binding results in constitutive expression of the lac genes. They are permanently on, regardless of lactose. Framework 2. DNA polymerase III proofreading is carried out by the epsilon subunit, using its 3' to 5' exonuclease activity. It excises misincorporated nucleotides co-replicatively. This is distinct from post-replicative mismatch repair. Framework 3. Lagging strand synthesis produces multiple Okazaki fragments. Each fragment requires its own RNA primer synthesized by primase. Fragments are later ligated into a continuous strand. Framework 4. RISC containing siRNA cleaves target mRNA at the position complementary to nucleotides 10 and 11 of the guide strand. Argonaute 2 is the slicer. Framework 5. Insertional inactivation of the antibiotic resistance gene, not the reported gene, means bacteria with the plasmid cannot grow on selection plates. No growth, no colonies, no color readout. Each of these frameworks is a class of reasoning. If you understand why constitutive expression follows from operator disruption, you can answer any Operon regulation variant. If you understand the geometry of lagging strand synthesis, every Okazaki fragment question becomes trivial. This is the GEK JRF Academy approach. One mechanism understood completely eliminates an entire category of exam questions. Practice now at gekjrfacademy.com.